Chapter 2, Basic Chemistry. Here are your learning objectives for this chapter. Just a quick disclaimer. Chemistry was never my strong suit, so while I will have some candor and some add-ons to the material in the book, a lot of it is going to be directly from your text because I don't want to get anything too muddled for you. So our learning objectives: differentiate matter from energy, list 4 major energy forms and provide one example of how each is used in the body, define element and list the 4 elements that form the bulk of body matter, explain how elements and atoms are related, define molecule and explain how molecules are related to compounds, recognize that chemical reactions involve the interaction of electrons to make and break chemical bonds, differentiate between ionic, polar covalent, and nonpolar covalent bonds, and describe the importance of hydrogen bonds, contrast synthesis, decomposition, and exchange reactions, distinguish organic from inorganic compounds. What's the matter? With some exceptions, matter can be seen, smelled, and felt. It's anything that occupies space and has mass, and weight is the measure of gravity pulling on that mass. So in the human body, the kinds of matter we have are solids, liquids, and gases. Matter itself can be changed both physically and chemically. Physically, it doesn't alter the basic nature of the substance, for example, ice melting into water or food being cut up into smaller pieces. And then when it's altered chemically, it does alter the composition. So this would be like fermenting grapes for wine, digesting food. So in our body, solids, there's bones and teeth, define shape and define volume. Liquid, like blood, plasma, or interstitial fluid, define volume but conforms to the shape of its container. And then gas, the air you breathe, no defined shape, no defined volume. Energy. So energy doesn't have mass, has no mass, does not take up space. Instead, it is measured by the effects it has on matter. It is the ability to do work or to put matter into motion. All living things are built of matter, and to grow or function, they require energy. So matter is the substance and energy is the substance mover. Here, we're going to talk about kinetic and potential energy. So kinetic energy is energy, quote unquote, doing work, aka putting that matter into motion. And you can see this on the atom level or just with your plain eye. Atoms moving or a bouncing ball. Potential energy is that inactive or stored energy, for example, batteries in an unused toy. So all forms of energy exhibit both kinetic and potential work capacities. Here let's talk about chemical and electrical energy. Chemical energy is stored in the bonds of chemical substances. The potential energy is in those chemical bonds, for example, gasoline molecules, and the kinetic energy is when the bonds and molecules are broken down. And for gasoline, when they release that energy to power your vehicle. Electrical energy results from the movement of charged particles. So you can see the example here is just wiring in your home. That flow of subatomic particles moves along the wiring. In yourself, your nervous system uses the electrical currents called nerve impulses to transmit messages. Those charged particles are called ions. Now let's talk about mechanical and radiant energy. So mechanical energy is kind of what it sounds like. The mechanics directly involved in the moving of matter. So your example here is the muscles in your legs shorten, contract, they pull on your bones, enabling your limbs to move and enabling you to, let's say, pedal a bike or walk down the street. Radiant energy travels in waves, and it's of the electromagnetic spectrum. So this would be like light energy stimulating your retinas which enables you to see, or UV waves causing sunburn, but also being able to stimulate vitamin D production in your body. If any of you ever took high school chem, you probably recognize this, the periodic table of elements. So all matter is composed of a limited number of substances called elements. Here are 118 that have so far been identified with certainty. 92 occur in nature, while the rest are artificially made in accelerator devices. Listed here are the 4 elements that make up about 96% of the human body: oxygen, carbon, hydrogen, and nitrogen. Oxygen comes in at 65%. It's a major component of organic and inorganic molecules. As a gas, it's essential for oxidation of glucose and other food fuels, during which cell energy is produced. That cell energy is called ATP, which we'll learn about later in the lecture. Then we have carbon at 18.5%. It's a primary element in all organic molecules. Examples of organic molecules include carbohydrates, lipids, proteins, and nucleic acids. Then we have hydrogen, making up about 9.5%. This is also a component of all organic molecules. And then coming in last, nitrogen, 3.2%. It is a component of proteins and nucleic acids, which are genetic materials. Each element is composed of similar particles called atoms, but all are unique. And you can see their designations here, O, C, H, and N. Each element has a designation of 1 or 2 letters called the atomic symbol. Here you can see the other trace elements in the body: calcium, phosphorus, potassium, sulfur, sodium, chlorine, magnesium, iodine, and iron. Again, just note their atomic symbol there on the side. You can see where some of them have the 2 letters. Calcium has Ca, and some have 1. Potassium has K. Now let's talk about atomic subparticles. So first of all, the word atom comes from the Greek word meaning incapable of being divided. We have protons, neutrons, and electrons. So protons and neutrons can both be found in the nucleus. They both have a mass of 1. Protons have a positive charge and neutrons have a neutral charge. Electrons orbit around the nucleus, and the mass of the electron is actually so indiscernible that we just assume it to be 0, and it has a negative charge. Electrical charge of a particle is measured by its ability to attract or repel other charged particles. All atoms are electrically neutral, meaning that they have the same number of protons and electrons, which cancels the charge out. Helium has 2 protons and 2 electrons. Iron has 26 of each. So atoms that have lost or gained electrons are then considered ions. So you can say here on the right, in the nucleus in the center, you have 2 neutrons and 2 protons, and then going around the periphery, you have your electrons. The image here on the left is similar to the image you saw on the previous slide. It demonstrates a planetary model of an atom, which is considered outdated, but it's simple and it's a good way to learn and understand. So you can see the atomic nucleus is made up of neutrons and protons. It is dense, it's positively charged, and then the electrons move in an orbit around that on the general location of the outside of the nucleus. You can see her in the same image. The right atom has a haze, an electron haze, considered the electron cloud. All atoms have protons, neutrons, and electrons. So what differentiates them is the numbers of each. The number of neutrons can be difficult to determine. However, as a general rule of thumb, lighter atoms tend to have equal numbers of protons and neutrons, while the larger atoms have more neutrons than they have protons. We use atomic number, atomic mass number, and atomic weight to really identify particular elements. And we'll talk about that on the next slide. First, we have the atomic number. This is the number of protons an atom contains. It's a unique number and will indirectly tell you how many electrons an atom has. Because if you remember, atoms are electrically neutral, so it has to have the same number of protons and the same number of electrons. Atomic mass number, aka mass number, is the sum of the masses of all the protons and neutrons contained in the nucleus. For example, hydrogen has 1 proton and no neutrons, so its atomic mass number is 1, shown in subscript to the left of the atomic symbol. And then we have atomic weight and isotopes. Atoms of almost all elements come in variations called isotopes. They have different numbers of neutrons, which will add variation to their mass numbers. Isotopes of an element have the same atomic number, so they have the same number of protons, but different atomic masses because they have variable number of neutrons. As a general rule, the atomic weight of an element is approximately equal to the atomic mass number of its most abundant isotope. I know this is getting kind of confusing, but just try to stay with me here. Heavier isotopes of certain atoms are unstable, and they release energy to become more stable. These are called radioisotopes. The process of spontaneous atomic decay, in other words, turning isotope to radioisotope, is called radioactivity. Radioactive decay involves alpha or beta particles or gamma rays from the atom's nucleus and are damaging to living cells. So alpha particles are the least penetrating, while gamma are the most penetrating. An example of something real world that uses radioisotopes is a PET scan. A radioisotope of iodine can also be used to scan the thyroid gland of a patient who is suspected of having thyroid cancer. Radium, cobalt, and other radioisotopes are used to destroy cancer. So ionizing radiation does not damage atoms in its pathway directly. Instead, it sends electrons flying, which do the damage. They're used in minute amounts to tag biological molecules so that they can be followed through the body. So that would be how they're used in imaging. So when you add 2 or more atoms together, it makes a molecule. If 2 or more atoms of the same element are bonded or become linked, a molecule of that element is produced. So here we have a hydrogen atom and a hydrogen atom coming together to make a hydrogen gas molecule. So the chemical equation would be the reactants, aka the atoms, are indicated by their atomic symbols, so H plus H, and the product, aka the molecule that is formed, the hydrogen gas molecule, is indicated by the molecular formula that shows its atomic makeup. So its atomic makeup is 2 H's, so H subscript 2. Compounds. When 2 or more different atoms bind together to form a molecule, this is called a compound. So 4 hydrogen atoms and 1 carbon atom interact chemically to make methane. So you see here again, you have the atomic symbol 4H plus C, and that turns into CH subscript 4, because there's 4 H's and 1 C. Compounds have characteristics that differ greatly from the atoms that make them up. Example would be table salt. You can see you put sodium, which is a silvery metal, and chlorine, which is a poisonous gas, together, and we get salt. Chemical bonds and chemical reactions. So chemical reaction occurs when atoms combine with or dissociate from other atoms chemically. So chemical bonds are formed. A chemical bond is not an actual physical structure, but it is rather an energy relationship that involves the interactions between the electrons of the reacting atoms. The role of electrons. So electron shells or energy levels are the orbits. They generally stay in fixed regions of space around the nucleus. The valence shell is the atom's outermost shell. Its valence electrons determine the chemical behavior of the atom. So there's something called the rule of eights. In the absence of a full valence shell, atoms interact in such a way that they will have 8 electrons in their valence shell. So when the valence shell of an atom contains 8 electrons, it is inert or stable and is chemically inactive. Fewer than 8 electrons, an atom will tend to gain, lose, or share electrons to reach a stable state. Any of these occur and chemical bonds are formed. Here at the top of the image on the right, you can see examples of chemically inert elements. So helium, well, it doesn't have 8, it only has 2, but that's all it can hold. So it's considered full. And the neon does have 8 in that outermost ring, that valence orbit. And then down below, you can see some of them have 4, 1, 6. So they just really desire to have 8 on the outside. And so they will find another atom or molecule and they'll connect to try to get that 8. Ionic bonds. So these form when electrons are completely transferred from one atom to another. An atom gains an electron and is now a negatively charged ion called an anion, indicated by a minus sign. If an atom loses an electron, now it's positively charged and it's called a cation ion, indicated by a plus sign. So here on the right is an example of ionic bonding, the formation of salt. So you can see sodium is quote unquote gifting the chlorine atom an electron, and then they both have 8 on their outer shell, their outermost ring, valence shell, and it becomes sodium chloride. Covalent bonds. So these molecules have atoms that share valence electrons rather than gift valence of electrons. Co with valent having power shared electrons. So at the top you see 2 hydrogen atoms joining electrons, little buddies, and it turns into a molecule of hydrogen gas. Then you see just below that double covalent bonds. You see two pairs of oxygen electrons joining together, becoming a molecule of oxygen gas. And then below that we'll see formation of 4 single covalent bonds. The 4 hydrogens are going to link with the 4 electrons on the carbon and become a molecule of methane gas. talking more about covalent bonds, we have nonpolar versus polar molecules. So just looking at these pictures, see if you can figure out which one is considered a polar molecule. Carbon dioxide is formed when the carbon atom shares its 4 valence electrons with 2 oxygen atoms. So the carbon is the gray atom in the middle, and the 2 red ones on the side are the oxygen atoms. Oxygen is very electron hungry, so it attracts the shared electrons much more strongly than the carbon, but because carbon dioxide is a linear molecule, the electron pulling power of one oxygen atom is offset by the other, so it's like an equal tug of war. So that is considered a non-polar molecule. If you see the water at the bottom, you can see that those hydrogen molecules are pulling towards the bottom. They're pulling towards a pole. That is a polar molecule. So it's formed when 2 hydrogen atoms bind covalently to a single oxygen atom. Each hydrogen atom shares an electron pair with the oxygen atom, and again, the oxygen has a stronger electron attracting ability, but in this case, the molecule is formed as a V-shaped. The 2 hydrogen atoms are located at one end of the molecule, and the oxygen atom is at the other. Consequently, the electron pairs are not shared equally. This arrangement allows them to spend more time in the vicinity of the oxygen atom. Because the electrons are negatively charged, that end of the molecule becomes slightly more negative, which is denoted here on the image with the little minus sign, and the hydrogen end becomes slightly more positive, noted by the positive sign. In other words, a polar molecule, so a molecule that has 2 charged poles, is formed. So let's talk about hydrogen bonds. They're extremely weak, and they're formed when a hydrogen atom bound to one electron-hungry nitrogen or oxygen atom is attracted by another such atom, and the hydrogen atom forms a quote-unquote bridge between them. Electrons are not involved in this. It's common in water molecules and important in intramolecular bonds, and it helps build different parts of the same molecule. It's important in helping maintain structure of protein molecules and DNA. And in the image here, you can see in image A, the slightly positive ends of the water molecules become aligned with the slightly negative ends of the water molecules. And then in image B, you see this little bug. The water's high surface tension is a result of combined strength of its hydrogen bonds, and it allows the water strider, the bug, to walk on the pond without breaking the surface. Pattern of chemical reactions. We have synthesis reactions, decomposition reactions, and exchange reactions. So synthesis reactions occur when 2 or more atoms or molecules combine to form a larger, more complex molecule. A plus B becomes AB. They always involve the bond formation, energy storing reactions, and it underlies all anabolic activities that occur in body cells. So building, example would be amino acids. Decomposition reactions occur when a molecule is broken down into smaller molecules, atoms, or ions. So AB turns into A plus B. So it's basically a synthesis reaction in reverse. Bonds are always broken. The products of these reactions are smaller and simpler than the original molecules. Chemical energy is released, and it underlies all catabolic processes. Example, the digestion of food and the breakdown of glycogen. Exchange reactions involve simultaneous synthesis and decomposition reactions. Bonds are made and broken, as you can see denoted by the lettering right below it, the formula right below it. So the example of this would be ATP reacts with glucose and transfers its end phosphate group to glucose, forming glucose phosphate, and becomes ADP. So again, we'll talk about this later in the lecture, ATP to ADP, and then ADP back to ATP. This image is straight from your text and is just breaking down synthesis reactions, decomposition reactions, and exchange reactions again. So in the synthesis reaction, you see that small particles are bonded together to form large, more complex molecules. So the amino acid molecules link together to become a protein molecule. In the decomposition reaction, the example they have is glycogen. So glycogen is broken down into glucose molecules. And then exchange reaction. So you have here Glucose plus your ATP. That little end P, the end phosphate group, breaks off and attaches to the glucose to become glucose phosphate, and the ATP turns into ADP. So it goes from triphosphate to diphosphate. So factors influencing the rate of chemical reactions. We have an increase in temperature, which will then increase the kinetic energy, so that movement energy of molecules, which in turn make them move more rapidly and have them collide more frequently. An increased concentration of reacting particles. Obviously, if you have more things in one place, there's more chance of them being bumping into each other because they're crowded together. A decrease in particle size, so smaller particles have more kinetic energy and move faster than larger ones, hence they take part in more collisions. And then the presence of catalysts decrease the amount of energy the molecules need to interact by holding the reactants in the proper positions to interact. So this is really things that influence an increased rate of chemical reactions. So now let's get into what composes living matter. We have inorganic compounds and organic compounds. So inorganic compounds lack carbon. They tend to be small and simple molecules. There are some unexplainable exceptions, like carbon dioxide and carbon monoxide, but examples would be water, salts, and many acids and bases. And then organic compounds. They contain carbon, carbohydrates, lipids, proteins, and nucleic acids, and they're fairly or very large covalent molecules. So the first inorganic compound we're going to talk about is water. So it is the most abundant inorganic compound in the body. Your body's made about 60 to 80% of water. So it has a couple properties: high heat capacity, polarity and solvent properties, chemical reactivity, and cushioning. So water absorbs and releases large amounts of heat before it causes temperature change. So this will help prevent your body from going into sudden temperature changes. It's considered a universal solvent. So it's a liquid. Solvent is a liquid or gas in which similar amounts of other substances called solutes can be dissolved or suspended. Tiny particles are a solution, large particles are suspension, and intermediate-sized particles are considered colloids. Chemical reactions in your body depend on water. So water molecules are added to the bonds of larger molecules to break them. This is called hydrolysis. Water also acts as a transport and exchange medium in your body. Things like mucus and synovial fluid, which is for all intents and purposes joint oil, use water as their solvent. It's also protective. So if you think about your brain, the CSF needs that water so your brain is floating and it's nice and cushioned. Salt is another inorganic compound found in your body. It's an ionic compound containing cations other than the hydrogen ion and anions other than the hydroxide ion. Salts containing calcium and phosphorus are found in the bones and the teeth. When they're dissolved in water, salts can easily separate into their ions. They're vital to body functioning. And charged particles, so all are electrolytes or substances that conduct an electrical current in solution. So in the image on the left, you can see the slightly negative ends of the water molecules are attracted to the positive sodium, whereas the slightly positive ends of the water molecules are oriented towards the negative chloride, causing the ions of the salt to crystallize and pull apart. Let's talk about the last category of inorganic compounds, acids and bases. So both are electrolytes and dissociate in water and can conduct an electrical current. So acids taste sour, can have really devastating effects. substances that can release hydrogen ions in detectable amounts. Proton donors. Strong acids would be, an example of that would be hydrochloric acid, and a weak acid, acetic or carbonic acid. Bases taste bitter, feel slippery. They're proton acceptors. An example would be hydroxides. Ionizes and dissociates in water, but the hydroxide ion and some cations are released. So a strong base is any base containing a positive hydrogen ion, and a weak base an example of that would be a bicarbonate ion. Acids, bases, and neutralization. So acids and bases are mixed forms of water and a salt. This type of interaction is called a neutralization reaction. pH, you can see the pH scale here on the right, shows relative concentration of hydrogen and hydroxide ions in various body fluids. So the pH scale is based on the number of hydrogen ions in the solution. Above 7 is basic or alkaline, and below 7 is acidic. So you can see here at the bottom, hydrochloric acid has a pH of 0, so it is highly, highly, highly acidic. Right here in the middle, you'll have things like blood, milk, egg whites are pretty close to middle. And then all the way in the other end, pH of 14, you'll have sodium hydroxide. Very basic. Now let's move on to our organic compounds. So these are very large molecules, but their interaction typically involve only small reactive parts of their structure called functional groups. So acid groups, amines, and others. Polymers are chain-like molecules made of many similar or repeating units, monomers. These are joined together by dehydration synthesis. So when polymers must be broken down, so going the other way, or digested to their monomers, the reverse process is called hydrolysis. So you see hydration or dehydration and hydrolysis. So you already know water is involved. During dehydration synthesis, a hydrogen atom is removed from one monomer and a hydroxyl group is removed from the monomer it is to be joined with. A covalent bond then forms, uniting the monomers, and a water molecule is released. Water is removed, dehydration. In hydrolysis, a water molecule is added to each bond. The bond is broken, releasing the monomers. These processes occur to all organic molecules discussed in this chapter, including carbohydrates, lipids, proteins, and nucleic acids. Carbohydrates. So this is an organic compound. Monosaccharides, disaccharides, polysaccharides. So you already see the base of the word is the same, and what's changing is the prefix. Mono, di, poly. Mono, one, di, two, poly, many. So monosaccharides are simple sugars, single chain or single ring structures. In the body we have glucose, which is aka blood sugar. It's a universal fuel. Fructose and galactose, which are converted into glucose, ribose, and deoxyribose, part of the structure for nucleic acids. Disaccharides, double sugars, two simple sugars joined by dehydration synthesis. You have sucrose, which is glucose and fructose together, which is cane sugar. Lactose, glucose and galactose together, found in milk. Maltose, glucose with another glucose, malt sugar. And in order for these to be digested, they must go through hydrolysis, so water added. Polysaccharides, many sugars, large insoluble molecules, ideal storage products, aren't very sweet. In the body, it's starch, grains and root vegetables, and glycogen, which is found in animal tissue. This is just a picture representation of what we just talked about in the last slide. So you have A, monosaccharide, single sugar, B, disaccharide, double sugar. You can see how dehydration synthesis will take the glucose and fructose and turn it into sucrose and water, and then you go back via hydrolysis, and it turns it back into glucose and fructose. And then D, you can see starch. It's a polysaccharide. There are many in a chain. It's important to remember that carbohydrates provide a ready, easily used source of fuel for cells, and glucose is the cell's favorite. Lipids, aka fats, are a large group, a diverse group of organic compounds. They enter the body in the form of meat, egg yolks, dairy, oils. The most abundant are triglycerides, phospholipids, and steroids, which we'll go a little more into on this slide. And like carbs, they are comprised of carbon, hydrogen, and oxygen, but carbon and hydrogen far outweigh the oxygen in lipids. Lipids are insoluble in water, but dissolve in other lipids and organic solvents like alcohol and acetone. So triglycerides, they're neutral fats, 3 fatty acids plus 1 glycerol. So it's kind of E-shaped like a fork with 3 tines. Examples would be saturated fat, which is butter. Single covalent bonds between carbon atoms, so it's very tightly packed. Unsaturated, monosaturated, and polyunsaturated contain 1 or more double bonds, and those would be your oils. Trans fats are oils solidified by the addition of hydrogen. And then you have things like omega-3 fatty acids, which will be found in fish. Phospholipids, a phosphorus-containing group. The head takes the place of one of the fatty acid chains. The head is negatively charged and has special chemical properties and polarity. The charge region, the head of the phosphorus, is hydrophilic, and the fatty acid chains are hydrophobic. So hydrophilic, water-loving; hydrophobic, water-hating, repels water. This allows cell membranes to be selective about what gets in and out. So cell membranes are made up of phospholipids. And then steroids. These are flat molecules formed by 4 interlocking carbon rings. They're fat soluble, made primarily of carbon and hydrogen. And how it exists in the human body is cholesterol. So cholesterol is generally a bad word. You hear it a lot when it's talked about in heart health. However, it is essential for life. It's also the raw material for making things like vitamin D, your steroid hormones, and bile salts. The next organic compound, proteins. So this accounts for over 50% of organic matter in the body. It contains hydrogen, oxygen, and carbon, just like the others, but plus nitrogen and sometimes sulfur. So small molecular building blocks are called amino acids, and there are 20 different amino acids found in the human body. All have an amine group, an acid group, and a different R group. So your R group is what's going to differentiate the different functions of the amino acid. So you can see here on the right, there's 5 different ones, and you can see they all have in common the base of the amine group, the acid group, and then they all have a different R group. So all amino acids have both the amine and the acid, and they differ only in atomic makeup of their R groups, specific structures of the 4 amino acids. So that's what you're going to see in B through E: glycine, aspartic acid, lysine, and cysteine. Proteins are already also segmented further. Polypeptides are fewer than 50 amino acids joined together in a chain. Proteins are more than 50, and large complex proteins are 50 to thousands of amino acids. So not only does each amino acid have a unique function, depending on the order that they are linked together in that chain, it will further define the function of that protein. Structural levels of proteins. So we have primary, secondary, tertiary, and quaternary. So primary structure, protein's primary structure is the unique sequence, just like I said, of the amino acids in the polypeptide chain. Secondary is going to be the alpha helix and the beta pleated sheet. It's reinforced by hydrogen bonds and represented by dashed lines in this figure. The tertiary structure is the overall 3-dimensional shape of the polypeptide protein. It's reinforced by chemical bonds between the R groups of the amino acids in different regions of the protein chain. And quaternary. Some proteins consist of 2 or more peptide chains. For example, the 4 peptides construct the protein hemoglobin, a blood protein. There are fibrous and globular proteins. So fibrous are structural, things like collagen, and globular are functional proteins, things like hemoglobin. Other functional proteins in the body include antibodies, hormones, transport proteins, and enzymes. We're going to talk a little bit more about enzymes just because they are so essential to body functioning. Enzymes and enzyme activity. Enzymes are functional proteins that act as biological catalysts, and catalyst being a substance that increases the rate of a chemical reaction without becoming part of the product or being changed itself. Enzymes can accomplish this feat because they have unique regions called active sites. These sites fit, quote unquote, fit and interact chemically with other molecules of complementary shape and charge called substrates. While the substrates are bound to the enzyme's active site, producing a structure called an enzyme substrate complex, they undergo structural changes that result in a new product. Whereas some enzymes build larger molecules, others break things into smaller pieces or simply modify a substrate. Once the reaction has occurred, the enzyme releases the product. Because enzymes are not changed during the reaction, they are reusable, which is pretty cool, and the cells need only small amounts of each enzyme. Think of scissors cutting paper. The scissors are the enzyme, and they remain unchanged while the paper, the substrate, is cut to become the product. The scissors are reusable and go on to cut other paper. So this image here is just a simplified view of enzyme action. So you have the substrate, so simple sugars, and the active sites on the enzyme becomes the enzyme substrate complex. Energy is absorbed. The bond is formed by dehydration synthesis, so you see that little water molecule coming off, and then it creates the product, which just goes on its merry way, and we still have the enzyme left there and with the ability to continue keeping on. So it can do this whole process again without change to itself. They're capable of catalyzing millions of reactions each minute. However, they do more than just increase the speed of chemical reactions. They can also determine which reactions are possible at a particular time. No enzyme, no reaction. Enzymes can be compared to a bellows used to fan a sluggish fire into flaming activity. Without enzymes, biochemical reactions would occur far too slowly to sustain life. So there are hundreds of different kinds of enzymes in the body, and they're very specific in their activities. Each controls one or a small group of chemical reactions and acts only on specific substrates. And most are named according to the specific type of reaction they catalyze. So for example, hydrolysis, HYDRO, which should already tell you something, LASES, add water, and oxidases cause oxidation. In most cases, you can recognize an enzyme by the suffix ASE, ACE, in its name. Now let's move on to nucleic acids. So nucleic acids make up your genes. It's the largest biological molecule in the body, composed of carbon, oxygen, hydrogen, nitrogen, and phosphorus. Considered the building blocks, so a nitrogen-containing base, a pentose sugar, and a phosphate group. The bases come in 5 varieties. So we have A, G, C, T, U, adenine, guanine, cytosine, thymine, and uracil. The 2 major kinds of nucleic acids are deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. So you can see here represented something that should look pretty familiar to you, a diagram of the DNA molecule. So the subunit of DNA, the nucleotide, is composed of deoxyribose sugar molecule linked to a phosphate group. A nitrogen-containing base is attached to the sugar, and the nucleotide illustrated both in its A, so you can see A, adenine nucleotide, chemical, and B, diagrammatic structures. Adenine nucleotide contains the base adenine. C is the computer-generated image of a DNA molecule, and D is the diagrammatic structure of a DNA molecule. So you can see the two nucleotide chains coiled into a double helix. The backbones of DNA are formed by alternating sugar and phosphate molecules. The rungs, so it's almost like a little ladder, are formed by complementary bases, A to T, G to C, bound by 2 or 3 hydrogen bonds, respectively. Adenosine triphosphate, ATP. So this is pretty important to just your body cell and your body functioning. It has been mentioned a few times in this lecture and in your text prior to them really explaining it, prior to me really explaining what it is. So it provides a form of chemical energy that all body cells can use, and it's a modified RNA nucleotide, which is why it's grouped with the nucleic acids. It has 3 phosphate groups instead of 1, triphosphate. Without it, molecules cannot be made or broken, cells cannot maintain their cell membrane boundaries, and all life processes stop. So what happens is glucose is oxidized, so think about where we get glucose from. Energy is released from that reaction, and it's captured and stored in the bonds of the ATP molecules, and they become these small packets of energy. And those small packets of energy is used to provide cellular energy, and then ADP starts to accumulate. So we're going from a triphosphate to a diphosphate. So think about what's happening here to change this name. We're losing a phosphate group. When the energy is being used, we're losing a phosphate group. It's becoming ADP. But we want to restore the ATP so these processes in our body can continue on. In order to go back from ADP to ATP, it's the oxidation of food fuels, and that's where glucose becomes oxidized again to capture and store that energy in the bonds of the ATP molecule. We have a couple images on this slide. The one on the left, you can see the structure of the ATP in A. You see the adenine base, the ribose sugar, like it is in all nucleic acids, but 3 phosphate groups, high energy phosphate bonds. Below that, you see the hydrolysis of ATP. So it's going through this process, it's going through hydrolysis, water is being added, it's losing that phosphate group, so it's now a diphosphate, and energy is being expelled. In the image on the right, it's 3 examples of how ATP drives cellular work. The high energy bonds of ATP release energy for use by the cell when they are broken. ATP is regenerated. Phosphate is again bound to ADP, making it ATP, as energy is released by the oxidation of food fuels and captured in a 3rd high energy bond. So in the first one, we have chemical work. ATP provides the energy needed to drive energy absorbing chemical reactions. So you have ATP, it sees A with a phosphate group, B becomes A, B, and kicks that phosphate group off and becomes ADP. Transport work. ATP drives the transport of certain solutes, amino acids, for example, across cell membranes. So you can see why this is very important in this reaction. Mechanical work. ATP activates contractile proteins in muscle cells so that the cells can shorten and perform mechanical work. ATP is added to relaxed muscle cell, acts on a relaxed muscle cell, becomes a contracted muscle cell. A phosphate is kicked off, and while energy is expelled, and it becomes ADP. Here's some critical thinking to do after you consume this lecture, read your text, for us to discuss during our Zoom meeting. So Sarah has a lower than normal amount of body fat. She tends to be chilly except on very hot days. Lauren has a higher than normal amount of body fat and tends to feel overheated most of the time. Explain the relative sensitivity to the environmental temperature of these 2 women on the basis of the information you've been given in the organic compound section of this chapter. So it's really just pointing you to the exact area you need to go look at to see how we might be able to explain on a chemical level why these 2 women experience perhaps the same environment differently when it comes to their own personal temperature regulation. Any questions, comments, or concerns, feel free to let me know. You can bring your questions to the Zoom meeting, you can add them to the discussion board on Blackboard, or you can send me an email. Thank you.